start the recording. Awesome. Okay, so we're going. So again, today we have a fantastic guest speaker, Dr. Christine Biermann, and we really appreciate, Christine, you taking the time to be with us today. And we're looking forward to your presentation. As you saw, Meron prepared a really nice poster that has been circulating. And some students are not going to be able to make it. I know several in my class, at least, that wanted to come, but they have other classes, other commitments, but they're looking forward to the recording. So I have the pleasure to briefly introduce and give you some background about Dr. Christine Biermann. Dr. Biermann is an assistant professor of geography and environmental studies at the University of Colorado Springs, Colorado Springs, and an affiliate assistant professor in geography at the University of Washington, where she was previously on the faculty of that university. Recently, she has edited the Handbook of Critical Physical Geography, which also features our very own Lisa Kelly, as well as Gregory Simon. So they are featured and they have contributions in this volume. Christine is also the director of the Tree Ring Lab at the University of Colorado Springs. And in the Tree Ring Lab, Christine is working on collaborative research that combines dendrochronology and remote sensing to investigate drought resilience in Colorado's lower montane forest, and a second project that analyzes the ecosocial effects of PFAS contamination in Colorado Springs. So her talk today is based on research funded by the National Science Foundation and material from an article that is forthcoming in the journal Science, Technology, and Human Values. The most fun, and this is, don't we all need something nice and fun? She mentions that the most fun she has had thus far in 2020 has not doing research, but was running the Pony Express Trail race around Rampant Reservoir in Woodland Park, Colorado. All right, so without further ado, Christine, thank you so much again, and we're looking forward to your presentation. And let me double check that you can share your screen. You should be able to share your screen now.
Thank you so much for that introduction, Raphael, and thank you all for having me. I will be bringing up my slides in just a moment. Okay, and just to verify, are you all seeing just the slide right now?
Yes.
Okay, great, thank you. Okay, I'm happy to be here. I'm happy to be sharing my research on the science and conservation of native and wild trout in the American West. My presentation today is Wild Trout Conservation, Restoration, and the Molecular Turn. And in our research, my colleague and collaborator, Dr. David Havlick and I explore how new genetic technologies like DNA sequencing are revolutionizing what we know about trout populations in the Rocky Mountain West. And in turn, this knowledge is radically changing how these populations are valued, protected, restored, and sometimes killed in the name of conservation. So over the past several years, I began kind of noticing what seemed like some pretty big changes underway in how trout are understood and managed across the US West. I began to notice, for example, a growing emphasis on genetically pure and native trout, and a whole slew of management projects seeking to eradicate hybrid and non-native trout populations, many of which were deliberately stocked as game fish just decades ago. And I also noticed a rethinking, a kind of grappling with the science of native cutthroat trout, as scientists began sifting through trout genetics and trying, sometimes in vain, to understand the evolutionary histories and the origin stories of particular fish. More and more, trout are being understood as genetic bodies. Their identities and their histories are viewed as functions of their DNA. And trout are not alone here. We see this with many other organisms, including people to a degree, where these organisms are thought of more and more as determined by genetics. And from these observations, some big questions began to rise to the surface. And I'll structure this presentation around two of these questions. First, what does it mean to think about trout as genetic bodies? How has this shift in thought occurred, and what does it mean for the scientific understanding of trout distribution and ecology? And then secondly, how are new molecular tools and technologies impacting trout conservation and management? Are they resulting in new management practices, new policies, new funding priorities? Is genetic information changing our ideas about what fish belong where? So let's start by thinking about how molecular tools and technologies have helped to bring about this new understanding of trout as genetic bodies. Before this rise in genetic technologies or this molecular turn, trout were identified by where they lived, what watershed, and by phenotype, by what they looked like, their observable physical characteristics, like spotting, coloration, numbers of fins or scales. But with the advent of better and cheaper and faster molecular techniques, trout genetics have been brought into sharper focus. Now trout are routinely subject to analyses that identify and classify them based on what seem like more objective metrics associated with DNA sequencing. So these technologies are introducing greater precision and standardization to fish identification, and so they seem to be clarifying these murky evolutionary lineages associated with trout. And I'll highlight two key tools that have become essential parts of the fisheries scientists' molecular toolbox. So the first, pictured here, is mitochondrial DNA. And mitochondrial DNA is genetic information encoded in the mitochondria of animals. And it evolves relatively quickly, and it's less complex than the DNA found in the nucleus of cells. So scientists can obtain a DNA sample, often from a small fin clip, like in the middle picture on this slide, and then can analyze the mitochondrial DNA sequences to reconstruct a phylogenetic tree that shows evolutionary relationships between different trout and different populations of trout, which is what you see on the right here. A second important tool in this molecular toolbox of fisheries scientists is environmental DNA. Now, this is genetic information or genetic material found in water and sediment or air samples rather than directly extracted from its biological source. So no fin clip needed, just a water sample. And organisms are constantly sloughing off material, sloughing off tissue and cells into the air or water around them, from feces, from molting, eggs, dead individuals, and more. So by taking a water sample, scientists can analyze environmental DNA and determine what types of organisms live in that body of water or that watershed. Now, this allows scientists to detect non-native, hybrid, or genetically impure fish in a watershed without ever encountering the organism as a whole. So with increasing ease and decreasing cost of these technologies, we're seeing this rapid proliferation of genetic data on what fish are found where and what the genetic composition or identity of those fish are. And this is data that's now available to wildlife managers. For most wildlife managers, the goal of using these new molecular tools and genetic information is to improve the conservation of biodiversity, especially of native or threatened species. So for trout, DNA analyses have actually determined that many of Western North America's native cutthroat trout, or what were considered native cutthroat trout, are in fact genetic hybrids with both cutthroat trout DNA and rainbow trout DNA, which rainbow trout is not native to the inland West. And with these new molecular tools, it's now possible to identify genetically pure organisms or populations, but on the other hand, also to be able to identify those organisms or populations that have hybridized or that aren't quite what scientists and managers believe them to be. So basically, this new ability to know a trout genetically allows managers to operationalize concepts like nativeness and genetic purity as management goals. the ability to evaluate the purity of an organism now spurs management recommendations. So even a trace of genetic mixing in some cases may be disqualifying or may warrant a population being removed or sentenced to death. So in other words, genetic science is now being called upon to help decide the fate of fish across entire watersheds. And this is true, as I mentioned before, for many other species as well. Trout are just one of many organisms for whom wildlife managers are making life and death decisions based on differences in genetic makeup or genealogy. Wolves, tortoises, bison, there are lots of examples of this turn toward framing organisms as genetic bodies. For trout, managers frequently target hybridized or non-native populations with toxicants and replace them with fish deemed native to a given watershed. Now, this is not itself new. Fish toxicants have been used for decades to poison and eradicate undesirable fish. But what's new here is that molecular analyses are contributing to new geographies of eradication and conservation. Streams and populations that were once conservation priorities in situ may now be targeted for removal, and vice versa. So we have this situation where genetic knowledge is shaking up the status quo of conservation and fisheries management. And managers are increasingly leaning on genetic analyses for the final word in management decisions. Now, interestingly, this is happening even as ecological and social scientists are critiquing the ambiguity and the racialized meanings of concepts and categories like native and pure. Before I get into some specific examples of how this new genetic information is altering conservation and management, I first want to consider how the scientific understanding of cutthroat trout distribution and diversity has evolved and the role of genetics in this evolution. So cutthroat trout are salmonid fish native to cold water environments of Western North America. And over millions of years, they have evolved through geographic isolation into 20-some subspecies, each native to a different watershed. And I say 20-some because the exact number of subspecies and the categories are fraught and unclear and contested. And some of these different subspecies or these different lineages are pictured on the left here. And on the right, we see one depiction of cutthroat trout native ranges, showing some of the different lineages occupying different drainage basins. Later on, I'll focus on some examples of greenback cutthroat, which is believed to be native to the South Platte watershed in Colorado, and also I'll talk about the Rio Grande cutthroat, which you see the range in purple, which is native to the Rio Grande basin. Okay, but over the past few decades, these maps and these classifications have been revised and questioned and revised and questioned time and time again. The distribution and diversity of cutthroat trout has long been clouded with uncertainty. And even to lifelong scholars of trout taxonomy, it's been incredibly difficult to distinguish one species or subspecies from the next. Often different subspecies of cutthroat trout look nearly identical. And even though different lineages evolved in different areas, we can no longer assume that trout living in different areas of the West are indeed different subspecies. There's been widespread stocking and movement of fish since the 1800s, and this is sometimes referred to as bucket biology, where people move fish from one place to another without fully understanding which fish they're handling, where those fish have come from, or how those fish would interact with other organisms once they arrived. Cutthroat trout especially have been relatively easy to transport and cultivate, and they've hybridized readily with rainbow trout and within and between subspecies. So by the mid-20th century, most cutthroat trout subspecies, as shown on this map, were found only in very small portions of their pre-settlement ranges due to a combination of habitat loss, fragmentation, hybridization, and competition with introduced trout. So we have this mixed up biogeography that's long been shrouded in uncertainty. And that's a challenge for managers, and it's made management difficult, to say the least. And the decline of one particular subspecies of cutthroat trout, the greenback cutthroat, which you may be familiar with living in Colorado, is Colorado's state fish. The decline of this subspecies was particularly dramatic. By 1919, just a handful of small isolated populations remained. And less than 2 decades later, by 1937, the greenback was believed to be extinct. But then in 1968, the leading authority on trout at the time, Dr. Robert Banke, apparently sometimes he was called the trout doctor. He's kind of legendary in the Colorado fish world. He identified what he believed was an isolated surviving population of greenback cutthroats in the South Platte River Basin. And he identified this population based on phenotype, based on its physical characteristics and its traits. Genetic tools and a genetic differentiation between different subspecies was not yet available in 1968 when he discovered this population. This discovery that the greenback cutthroat was not extinct led to the fish gaining early protection under the U.S. Endangered Species Act. The U.S. Fish and Wildlife Service developed the first recovery plan for the species in 1978, and their goals for greenback cutthroat trout recovery were to establish 20 stable populations occupying at least 50 hectares of lakes and 50 kilometers of stream. By the late 1990s, it appeared that these goals had almost been met. There were 18 stable populations in the South Platte and 3 in the Arkansas River watershed. The recovery program appeared to be a success, and conversations began about delisting the subspecies or removing the Endangered Species Act protection. Everything appeared to be going well. But just as the greenback was poised for delisting, a few scientists started raising deeper concerns about the robustness of greenback recovery. They wondered if enough was known about the greenback and its life history to adequately restore it. They questioned whether the habitat was right, whether restoration sites were big enough for the fish to develop their mobile life histories. They wondered if the greenback was interbreeding with other species. And so as these critiques mounted, researchers at CU Boulder began to analyze the genetic composition of greenback cutthroats using mitochondrial DNA. They also began to scour museums and archives looking for preserved specimens to compare to the greenback DNA of today. And what they found was surprising. First, they found that the museum specimen that had long been believed to be the greenback cutthroat trout had actually been mislabeled and probably came from the Rio Grande watershed. So they went back to the archives and eventually came up with what is believed to be a trout that was collected from the South Platte watershed, where the greenback is presumed native. Then, after comparing existing greenback populations to the DNA from museum specimens, they found that these populations that they believed to be greenback cutthroat trout, this organism that had been the focus of state and federal recovery efforts for more than 40 years, was essentially the wrong fish. Populations of greenback cutthroat that had been carefully reared, protected, and restored to front range habitats were more likely variants of a different subspecies, the Colorado River cutthroat trout that was found west of the Continental Divide. Now, this finding threw a wrench in state and federal management plans. It challenged these nearly 40 years of recovery efforts. And the lead author, Jessica Metcalf, the geneticist who led the study, did not mince words. She wrote, "Our results imply that more than 2 decades of work toward bringing the species back from extinction have failed to improve the species' status." The discovery that decades of recovery had been based on this case of mistaken identity proved disorienting to scientists, to wildlife officials, and to anglers who had been studying, managing, or catching these fish for years, myself included. This was a fish I caught in 2009 that I believed was a greenback cutthroat trout, only to find out later that it was probably not. In little more than a decade, this observation-based biology that had been guiding fisheries management began to face challenges by laboratory tests that could find tiny differences in lineage and genetic composition of trout and other organisms. So as a result, this scientific understanding of cutthroat trout has been reconstructed from one based in direct experiences with fish in particular places to more abstracted DNA analyses of fish tissue or environmental samples. Now, the same DNA analyses that had revealed those restored greenback populations as the wrong fish also discovered one remaining population of greenback cutthroat trout that did match the museum specimens used for historical reference. These pure greenbacks were finning obscurely in an inconspicuous drainage, Bear Creek, located high on the southeast slopes of Pikes Peak, importantly in the upper reaches of the Arkansas River watershed. These new analyses, though, also highlighted that the true native cutthroat trout of this Arkansas River watershed was probably not even the greenback, but rather the extinct yellowfin cutthroat trout. So basically, Bear Creek, outside of the historical range of the greenback cutthroat, was found to be home to the only documented population of genetically pure greenback cutthroat trout in the state. Now, this brings us to the second question that I outlined in the beginning. Once this genetic knowledge is produced, what do managers do with it? How do molecular tools and genetic information influence conservation and management? As scientists find themselves able to evaluate species identity and distribution through tests conducted at the molecular scale, they face questions about what to do with these insights. Now, this poses broad questions of policy. For example, how do we engage Endangered Species Act protection to respond to these changing taxonomies and revised notions of population size and location? This also raises questions about what to do with trout for whom wildness and nativeness are especially fraught. For example, like those Bear Creek greenbacks that are found outside of their native range, or fish that are genetic hybrids, or fish that are the results of previous rounds of restoration. So despite these complications, state and federal management agencies now routinely use genetic insights and tools to help guide what are often life and death decisions for trout populations. Phylogenetic analyses have led to the reevaluation of greenback cutthroat trout in Bear Creek. They used to be considered kind of oddities, and now they're considered prized genetic stock, subject to intensive protection, reproduction, and reintroduction efforts. But for other populations, genetic tests and their results can diminish their value. For example, I'll discuss an example from 150 kilometers south of Bear Creek near Great Sand Dunes, where genetic analysis led managers to target wild cutthroat trout for eradication due to fears and threats of hybridization. So these examples together, I'll talk about Bear Creek a bit more and then Sand Creek near the Great Sand Dunes. These examples illustrate how framing trout as genetic bodies can compel managers to either care for or kill trout populations in the interest of kind of fixing or rectifying decades of genetic disruption that people have played a large role in. Okay, so thinking about the Bear Creek greenback cutthroats, following the genetic tests that identified these fish as the only remaining pure greenback population, the right fish, but in the wrong watershed, scientists and managers have grappled with questions about what constitutes greenback recovery, how to manage the populations previously believed to be greenbacks, and how to weigh genetic purity versus long-term viability of the subspecies. Now, despite the realization that these fish were not likely native to the watershed, Bear Creek's trout have been protected in situ and subject to intensive efforts to preserve and propagate the subspecies. Starting with just 65 fish, managers at federal and state fish hatcheries in Colorado have now raised thousands of Bear Creek trout for recovery efforts. The very factors, though, that led to the preservation of this genetically pure population also threaten its viability. The source population of greenbacks in Bear Creek is small, approximately 800 fish occupying a really short and isolated stretch of headwater stream. The population likely experienced genetic bottlenecks during more than a century of isolation, and the progeny that have been raised in hatcheries for recovery and restocking have shown alarming rates of genetic defect and limited viability. As a result, scientists are intensively managing their reproduction using a technique called gene matrixing. Basically, it relies on an algorithm to match least closely related fish to improve their viability and survival. So in this picture, you see a specific greenback cutthroat trout is being removed from one of its numbered tanks to meet its match. And hatchery-raised progeny of these trout have now been stocked in several isolated streams, lakes, and reservoirs in the South Platte watershed. In some cases, they have come to replace the populations that were restored during earlier rounds of greenback recovery. To prevent these genetically pure Bear Creek greenbacks from hybridizing, any fish in these locations first must be removed. For example, West Creek in Rocky Mountain National Park was a site of previous misidentified greenback cutthroat trout restoration, and managers now face a dilemma about how to value these populations. These populations have been relabeled as Colorado River cutthroat trout, and so West Creek's fish have been removed via non-lethal electrofishing and relocated. Now, West Creek's fish are effectively lucky. Hybridized or non-native fish in many other locations have not been as fortunate and are eradicated with toxicants. Now, in some places, new barriers to fish passage must also be constructed to protect transplants from mixing with other trout in the watershed. The most ambitious greenback restoration effort is now occurring in the upper Cache La Poudre drainage in northern Colorado. And this project is massive. It is going to take up to 2 decades, and the goal is to create 37 miles of waters for the greenback. Now, this might not seem like a lot, but 37 miles of interconnected waters must then be kept disconnected from any other waters downstream. So, we can think of this in today's terms as kind of quarantining the fish. So natural barriers like the one pictured here, as well as engineered barriers, are used to prevent fish passage and to keep genetically pure fish genetically pure. Long term, the success of this effort to establish this pure native and eventually wild population, it rests on these repeated interventions to ensure the purity of the species at the molecular level. It also rests on interventions to ensure survival and reproduction. And it also rests on interventions to the landscape, significant modifications through barriers, for example. So this project, this largest native trout recovery effort in Colorado, this project that aims to naturalize and rewild the state fish in pure form. This project is produced through a series of carefully orchestrated human manipulations. This is not in itself a problem, but it illustrates the ways in which genetic knowledge is operationalized in ways that emphasize purity, nativeness, and naturalness. But to achieve these states, these projects require major human modifications and manipulations of the landscape and of the trout. A similar push for species purity is underway farther south in Colorado, where the Sand Creek watershed, as I mentioned earlier, is a focus of Rio Grande cutthroat management. So this is part of Great Sand Dunes National Park and Preserve in the Sangre de Cristo Mountains. And Upper and Lower Sand Creek Lakes, this is Upper Sand Creek Lake, pictured here, a really nice backpacking trip, are off the beaten path a bit. In a wilderness area, stunning views, relative solitude, and until about a month and a half ago, waters teeming with large cutthroat trout. These lakes and Sand Creek are not connected by surface flow to the broader Rio Grande Basin, so they're disconnected, they're isolated. And these lakes are believed to have been fishless prior to stocking. In the 1970s, the Colorado Division of Wildlife, now CPW, first brought fish into Sand Creek lakes. They stocked novel hybrids that they had bred in hatcheries, and these hybrids were mixes of Colorado River and Yellowstone cutthroat trout, which, I just love this, the Colorado Division of Wildlife called these hybrids, seemingly without irony, the Pikes Peak native, even though they weren't native to the area, nor were they even in the same watershed as Pikes Peak. And over the next few decades, since these fish were stocked, the agency kept stocking fish every now and then because they weren't quite sure how many fish were surviving over the winters. Then, beginning in 1998, the agency shifted strategies and priorities, and they began stocking the lakes with Rio Grande cutthroat trout, hoping to expand the range for native Rio Grande, the native Rio Grande subspecies. So this kind of happened as an increase in attention occurred toward native fish. This watershed was super attractive for native trout management because it was isolated, it was clean, cold water, and it was protected in a wilderness area within a national park unit. So seemingly a good place for long-term protection of a native subspecies. So by the early to mid 2000s, more and more effort went into promoting pure Rio Grande subspecies of cutthroat trout. The state really wanted, for one, to avoid the subspecies being listed under the Endangered Species Act. So they began looking for genetic refuges for these fish, where they would not be exposed to other cutthroat trout lineages or rainbow trout with whom they might interbreed. In 2019, pictured here, federal officials published a study that detailed the genetic composition of trout in Sand Creek lakes and streams. The figure you see here is from the study, and what it is showing is the genetic composition of individual fish sampled in Upper Sand Creek Lake. So each column is one specific fish. And what it shows is that the genetic composition, the majority of the genetic material aligned with the native Rio Grande cutthroat trout. That's the kind of bluish purple color. However, there was of hybridization. During the more than 4 decades of stocking, the fish in lakes had hybridized to include a mix of genetics from Yellowstone cutthroat trout, Colorado River cutthroat trout, and Rio Grande cutthroat trout, with a few rainbow trout.
Gotta get my earbuds.
In many cases, scientists were unable to see differences in the markings of the fish, even though genetic tests proved these fish to be rather different in origin. So 100% Rio Grande cutthroat, for example, looked nearly the same as a Rio Grande Yellowstone rainbow trout hybrid. Most anglers who visited the lakes knew nothing or little about the ancestry or purity of the fish they were catching, referring to cutthroat trout in our visits to the site as simply native and wild as well. They also knew that the fish in these lakes seemed to be thriving, many of the trout reaching 20 inches or more in length. But following this 2019 study, the NPS announced a plan to apply toxicants to remove all fish in the watershed in order to restore a genetically pure native Rio Grande cutthroat population. Of course, Rio Grande cutthroat trout are only native to Sand Creek in a very broad geographic sense, as the drainage is believed to have had no trout in it prior to human intervention. But the isolation, the hydrological isolation of this, these creek and these lakes made it an ideal prospect for maintaining this protected genetic refuge for Rio Grande trout. Although the region or the specific area has a large number of anglers and backpackers who visit the lake, concern about maintaining this genetic purity ruled the day. And eventually in September, just this past September, the watershed was treated with toxicants and all fish were eradicated. The next step is for managers to make sure that the lake is indeed devoid of fish, and then to restore the genetically pure Rio Grande cutthroat into the watershed. While in the Bear Creek case, if we compare the two, genetic analyses there compelled managers to care for and protect the population. Here, the genetic data worked against the fish, kind of dooming the fish to eradication. So again, genetic science is called upon to help decide the fate of fish across watersheds. And as one prominent trout writer, Ted Williams, tried to patiently explain to a reader who lamented the waste of killing off perfectly good trout, in a similar case, he wrote, "It would be great if it were possible to eliminate all alien and mongrel fish and at the same time use them, but it is not. The only way to save native fish from genetic introgression, competition, and predation, and resultant extirpation or extinction, is to poison out the mongrels and aliens with rotenone. That's a chemical toxicant. You need 100% kill, anything less and you fail. No other method works. So no, native fish recovery with rotenone is never wasteful." Now, what this quote illustrates is the genetic information that indicates that native fish are at risk of hybridization and introgression compels managers to eradicate fish using chemical treatments because there is a sense that no other method works, as Williams states. Now, returning to the questions that we began, the Bear Creek and Sand Creek cases help us begin to develop answers to these questions. In both the Sand Creek and the Bear Creek cases, information provided by genetic tests prevailed upon wildlife officials and effectively motivated them to move beyond their direct experiences with trout to take action based on genetic data. This is neither inherently good nor bad, but it is different from previous modes of trout management that relied upon direct experience and observational biology rather than molecular analyses. Also, these two cases illustrate that the new genetic knowledge produced through mitochondrial DNA and environmental DNA testing comes with consequences, comes with life or death consequences for the individuals and populations being analyzed. Tests demonstrating genetic purity now serve as critical managerial mandates, making these newly genetic bodies of trout subject to restoration efforts, sometimes terminal restoration efforts like we see in Sand Creek. Evidence of hybridization then requires the eradication of some organisms, or more accurately, entire populations, in order to bring forward genetically pure bodies. Molecular analyses reinforce control over fish populations and help justify and determine acts of care or killing by state and federal agencies. Fish management has long relied upon eradication of undesirable fish, and has long relied on human manipulation of ecosystems and fisheries. So this is not in itself new, nor is it inherently problematic, I would argue. Science can help us parse possible decisions, and genetic knowledge can indeed be very helpful. But what we also see happening is that the proliferation of genetic data is in a way depoliticizing these life or death management decisions, with managers pointing to the science as deterministic. Now, as Williams argued in the quote that I mentioned earlier, the only way to save native fish becomes to eradicate non-native, and genetically impure bodies using toxicants. This rhetoric of the only way avoids any conversations about scientific uncertainty, about lack of consensus among geneticists and scientists, about values or about ethics. In other words, so this shift to thinking about trout and, by extension, other organisms as genetic bodies is ushering in an era where management decisions are often reduced to questions of technology and purity, rather than accounting for diverse priorities of ecology, culture, ethics, economics, long-standing relationships between fish and people, or other forms of scientific knowledge like indigenous ways of knowing. While trout management has typically relied on human intervention, this turn to genetic science is bringing new lineages and subspecies into being. It's changing how we know fish. It's altering long-standing conservation priorities, and it's intensifying the manipulation of biological processes such as reproduction, dispersal. And as a result, and largely inadvertently, I would suggest, this emphasis on genetic science is pushing other social and ecological factors toward the margins of management decisions. These changes warrant greater conversation about the consequences of molecular analyses and the values embedded in trout science and conservation more broadly. And in addition to these broader concerns about genetic knowledge dominating and determining trout management, there are also more particular questions of technology and science. For example, since only short fragments of DNA from a handful of museum specimens are available to determine kind of our notion of our baseline knowledge of trout identity and distribution pre-European settlement, is this enough? How reliably or robustly can genetic tests establish the actual character of Colorado's native trout pre-settlement? For a population such as the Bear Creek greenback, whose genetic stock bottlenecked to just a few hundred populations, to what degree should genetic purity be preserved or should it be compromised in the interest of expanding the fish's range and promoting its viability? This question is far from hypothetical. As I noted earlier, the Bear Creek fish that's being raised in hatcheries is showing alarming rates of genetic defect and very limited viability. So some scientists are suggesting that this pure strain of cutthroat be intentionally crossed with the Colorado River cutthroat or another trout subspecies to create a recovery population, one that would not be genetically pure, but might have greater viability and higher survival rates over the long term. If we opt to protect genetically pure but fragile trout that require constant intervention to ensure their survival and viability, what does that do then to existing goals of supporting wild and native fish? Now, most of these questions remain far from settled, but the technology continues to evolve, and next generation genetic tests are analyzing organisms with ever greater precision. These advancements, though, seem likely to produce additional challenging questions rather than clearer resolution. It's also important to recognize that this molecular term, this increasing reliance on genetic information, reaches well beyond matters of trout and increasingly plays a role in conservation decisions globally. Conservation efforts are increasingly mediated and promoted through digital technologies, big data, and other innovations that turn human decision-making into what appears to be data-driven, automated outcomes. And this is what these cases lead me to express caution against. Trout have been actively managed for decades, but with this turn to genetic science, these fish are becoming even more intensively analyzed and manipulated. Again, not in itself a bad thing, but one that more conversation one that warrants greater conversation. So one of the big take-homes here is that there's a need to broaden conversations, and public knowledge about taken for granted concepts like nativeness and genetic purity in trout management. We increasingly see fisheries management leaning on genetic analyses for the final word, but perhaps there's a way to engage molecular analyses, to open up conversations about management and conservation rather than to preempt them. So in conclusion, thinking about trout as genetic bodies brings certain aspects of their identities into sharper focus. But in doing so, other complexities of ecology, of history, and management are often simplified or overlooked. The molecular turn has shifted questions about where trout belong to be addressed increasingly as a matter of DNA, which is used as an authoritative signal for nativeness and belonging. The shift toward thinking of trout as genetic bodies is inadvertently narrowing this field of vision, focusing management on questions of technology and purity, rather than engaging more diverse ecological, social, or cultural considerations. Thank you. I am happy to answer any questions, and thank you for this opportunity to present this work.
Fantastic. Thank you so much, Christine. Really interesting presentation. We really appreciate it. And yes, thank you for, that was just about to ask you if you can stop sharing. So yes, thank you so much. That was very, very interesting. So many points you brought up that I was like, yes, that's great. I was just remembering one student that I have in that class that I don't think he was able to make it today, but he was asking about the recording, and he will be at the edge of his chair because he's right into this thing of fisheries and precisely the analysis of genetics. So thank you so much. There are so many interesting points, but I will let the students and faculty, if you have any questions or points at this moment, please bring it up. If not, I'll take the microphone and don't let it go and ask my questions. So go ahead. Anyone that has a question, feel free to turn in your mic and pose the question, please.
Well, Christine, hi, my name's Aaron. Thank you so much for an amazing presentation today. I have a bunch of questions, but I'll just start with one in no particular order. I was just wondering what kind of pushback there might have been with regard to the, hopefully I pronounced this correctly, the rotenone. Is that how you pronounce the chemical agent? So with regard to applying a toxic agent to the geography in this case, what is there any sort of commentary on that you were able to learn that would be kind of noteworthy?
Yeah, thanks. Good to meet you, Aaron. So rotenone, it is a plant-based chemical that degrades rather rapidly once applied, and it is presumed to have no effect on humans, on mammals. It affects fish, and it affects aquatic invertebrates, insects, and within a couple of days, it is usually no longer present in the system. It has been used for several decades in fisheries management and is widely accepted in the fisheries management world. There is pushback from kind of outside the fishery, generally from outside the fisheries science and management communities. And that pushback, one thing I think is very interesting is often the response to that pushback is kind of an accusation of chemophobia, like, oh, that's just fear of any chemicals, and that's kind of related. There's a sense that that pushback is related to a lack of education among the public. I think it's very interesting the degree to which rotenone is accepted and seen as problem-free within the fisheries science and management community, though there are exceptions, because there, quite frankly, isn't as much known about it as, as certainly there could be. One thing I'm hopefully going to work on in the future actually is kind of like a chemical history of rotenone and its use in fisheries. I think it's fascinating. It's a chemical that used to be used in organic agriculture, but is no longer approved by the FDA for organic agriculture. So now it's marketed mainly as a pesticide. Hopefully that answers a bit of your question.
It does, thank you.
All right, any other questions? Again, just feel free to turn in your mic.
Oh, go ahead, Lisa, yeah.
Oh, sorry, Tom. Zoom is so uncomfortable with when you talk and how you do it. Thanks, though. Christine, I think this is one of my favorite kinds of talks, which is it was a story that left us with tons of questions, and I just loved it. So first, thanks. But so I think I got from the talk, right, we get from this like kind of situated knowledge of trout to this more abstract genetic knowledge. And somehow this reliance on these ideas of nativity in the wild in turn produce this highly engineered trout landscape that is somehow depoliticized because it's data driven. And I guess my question is, so if you got to repoliticize trout conservation and take that like 37 million, I guess like sort of, how do you embed this sort of molecular toolkit in a more humble trout science? Like what does that involve from your thinking around this issue, having been so steeped in it? Does that question sort of make sense?
Yeah, it does. It's just really hard. And I don't have the answers. So this project is at its beginning. So what it looks like for a kind of repoliticized trout management that engages different actors and different kinds of knowledges, you know, I don't have a clear sense of what that looks like. I have a few examples of ways in which I think it is being done in kind of very specific contexts. So one example would be the Salish Kootenai tribes in western Montana. They're they have native bull trout and native west slope cutthroat trout that they're very active in restoring and managing. And genetic science plays a huge role in their management practices. But it does so in the context too of long-term indigenous knowledge of the region, of the fish, kind of deep cultural ties and relationships between people and the habitats and the fish. And so the genetic knowledge in that case, I think, comes to help decision making, but in a way that does not remove kind of the broader context and the other connections between fish and people. So I see that case as one example of this type of management that certainly uses and relies upon genetic knowledge, but does not see that knowledge as deterministic or as helping or as, you know, being the only factor guiding decision making. But I'll have to think more about that too.
Yeah, that's a great start. Thanks so much.
Get there eventually.
Yeah, so Christine, thank you for a really great talk. My undergraduate degree is actually in fisheries biology, where I went to school up in Montana. That feels like a bit of a past life for me these days, but nevertheless, I thought it was fascinating and rooted in some really interesting science, and so really great in that regard. I'm kind of curious about your maybe your view on, so we haven't really talked much about climate change, for example, right? And the way in which the climate change is impacting freshwater ecosystems. And I wonder whether the kind of paradigm that we are in today, you know, thinking about kind of native species is almost, it's not a moot point, but it's certainly a challenging point kind of moving forward with increasing water temperatures and such. And I wonder whether you have any thoughts about maybe perhaps the next molecular turn, to use your terminology, right? Moving towards using genetics to identify kind of phenotypes, for example, that may be more well adapted to slightly increasing water temperatures, and whether that's like the next kind of the next kind of movement that we might be using, I guess, with these issues in mind, I suppose. Yeah.
Yeah, that's an awesome, fantastic point. And really, I probably should be interviewing you, Tom, I guess. But and that's what you mentioned, you know, using genetic knowledge and molecular tools to identify populations or specific lineages or traits that make a population more resilient to change. It's often associated with warmer water temperatures or just more fluctuating water temperatures. Scientists, especially those at University of Montana and the Forest Service's National Genomics Center in Missoula, are doing just that now, especially with the West Slope cutthroat trout in Montana. And I do think, as you said, I think that's kind of this next forefront of the molecular turn with fisheries and perhaps with wildlife conservation more broadly. And I think, you know, it's really interesting to me because that's an area we haven't focused a ton yet in our project, but my initial gut reaction is that that emphasis and having that as a decision-making criteria really helps this molecular turn in a sense, or helps politicize it in a way, because it means that the managers are coming in, are choosing what they're looking for and what they're hoping for, and how those choices are made, you know, that's kind of, who knows. But they aren't just, the genetic knowledge isn't just like truth, here is what we do. They're coming in with a kind of sense of what the goal is and using that knowledge to meet that goal. So I think more and more that is happening, but it's happening, you know, also I would say your experience in Montana and how that maybe shapes your views on this is interesting, because part of our project is comparing Colorado and Montana, and fisheries management in Montana is vastly different from fisheries management in Colorado. And in a way, the state of Montana and the researchers within Montana are kind of at the forefront and are pushing the boundaries of the ways molecular knowledge is being used. And Colorado is kind of playing catch up and almost coming in like where Montana maybe was a decade or two ago. So we see differences.
Yeah, I would suggest that a bit of that had to do with, you know, there were some really fantastic kind of fish geneticists at University of Montana for a very long time. Fred Allendorf, for example, who's unfortunately passed away now. But in any event, that I think that set kind of like a culture, if you will. And then the other giant difference up there, and we could talk about this offline, but essentially being the timber industry and the influence of the timber industry relative to that perhaps not being nearly the dynamic that's here in Colorado. But anyway, okay, yeah, great. Again, fascinating topic, and I like the work a lot. So,
thank you.
To kind of piggyback off of that question with climate change as well, I've been following a lot of like environmentalist and conservation organizations' efforts to rewild rivers with pushes to tear down dams on a lot of the rivers here in the American West. And I was curious to see what you or your research, if you believe that would be helpful, especially for these non-farmed fish populations, in order to mitigate like the drought and flood cycles that we have here in the American West.
Yeah, thanks for the question, Johnny. Certainly, I mean, the ecological havoc that dams can wreak on an ecosystem, you know, is well documented. And what's interesting when thinking about this kind of idea of promoting genetic purity of populations is this tension between connection and isolation. And in many cases, the genetically pure trout populations that remain in the West remain only due to human activities, which cut particular portions of a stream off from the broader watershed. So things like dam building, things like road and culvert building. So many of these genetically pure populations really remain. We have dams and roads and human activities to thank for their continuance on the landscape. And yet now there is this tension between wanting to promote habitat connectivity, especially in the wake of a changing climate, increasingly severe droughts, and promoting ecological, ecological processes across space, with this need also to isolate these populations. So, in the case of specifically preserving genetically pure trout populations, there's this tension.
Thank you.
Okay. Oh, go ahead, Kathy. Go ahead.
Gregory, did you have a question? Sorry.
I think Kathy has one. So.
Oh, sorry, Kathy. I didn't see you.
No problem. Yeah. Thank you, Christine. That was great. And to kind of follow up on the previous question, I was particularly interested in the discussion of kind of implemented barriers to these watersheds and almost mimicking, in some respects, geologic processes that, and I'm coming from this from a background of geoscience, and a lot of my initial interest in understanding fish was really around the speciesization of Pacific salmon and how linked that is to the geologic history of the Pacific Northwest and Alaska. Sort of as these habitats developed geologically, then the fish developed to use them. And I wonder if there's any sort of recognition at the management level that the geologic processes in watersheds have are always responsible for driving where species are, how populations or species diverge, and if that comes into this thinking about shutting off or making those fish barriers in different areas.
Yeah, that's a great question. And it's one I feel like I can't speak to it fully yet. It's something I probably should ask more when I'm doing interviews. In general, what I've experienced thus far is a sense that in terms of geology, what's focused on is really the long-term geological history of the West. So, like in thinking about Colorado's cutthroat trout subspecies, there is a huge emphasis on just the geological history and how that has shaped the migration and movement of these different subspecies to different areas. Or with the West Slope cutthroat trout in Montana, there's this, thinking about geology is only in the sense of thinking about, how did the fish cross the continental divide originally, how, that kind of thing. There's very little that I've experienced thus far with attempting to think through specifically thinking like about barriers or about how the current geological processes also are playing a role. But it's something that I would like to put on my radar more as I, from the 2 interviews that I've scheduled for next week.
Wonderful, thanks.
Hi, Christine. Thanks for that really great talk. And I just had a couple, comments and questions. And actually, it's interesting. I had a list of questions. And what's so great about this presentation is also the conversation we're having now, which has answered a lot of my questions, or I think we're all sort of thinking the same thing. But let me just first say that I really appreciate the kind of way in which you're engaging with both kind of the physical science world, but bringing sort of these social, theoretical perspectives on this, cultural perspectives. So I think for a lot of our students here, I think this is really emblematic of really good kind of work that blends these approaches. So I commend you for that. And I know you've done this work for a long time, but it just was a reminder that's really interesting. And I was also really struck by, I kept thinking every time you were talking about genetic purity, obviously my mind kept going to the sort of, questions of eugenics and like apartheid and separating out, and even this idea of sort of using those natural or man-made barriers, as you put it, quarantining as sort of forms of segregating, right? Biology based on genetic difference for the purpose of not letting them intermix and breed. So I just think all of that is really fascinating. There's sort of obviously a parable of sorts there. But I had a kind of a question that I'm not sure you'll be able to answer because it's about a different region. And I'm thinking of rainbow trout in California and how those actually are trout, but they become, and I'm maybe mixing up my fish knowledge here, not getting it right, but I think they become steelhead after a while of spending time in the ocean, then kind of come back and lay eggs in the rivers and streams, and just how that evolution of a species. So you have like rainbow trout, but they're actually trout and steelhead depending on what stage of life cycle they're in. And I'm just wondering if you could say anything about how people who are doing this work with fish that have that life cycle metamorphosis, how they deal with this genetic accounting. Because it seems like that's just an extra layer to have to consider when the fish itself actually changes over its life course.
Yeah, that's a good question. I mean, I think we could, thinking about rainbow trout and coastal rainbow trout, steelhead, which are native to the Pacific West Coast, though rainbow trout are not native to the inland West. I mean, it's similar because there's a coastal cutthroat trout species that does the same thing, has the same kind of life history, to the best of my knowledge. And, I think these questions of genetics are still, I don't think it necessarily changes because of that different life history in my sense so far. I'm not sure on that, but I'm thinking about salmon. I mean, and there are the same questions and debates and management practices around the genetics of salmon in the Pacific Northwest. And, so in a way I'm not sure that it's different because of that.
Yeah. I asked, there's an article that I called Species Complex written by a guy who I know, his name is Peter Alagona, and you may be familiar with this. It was in the Journal of Environmental History, and he kind of commented on how, steelhead are sort of this very protected species, and then you have rainbow trout, which is like hatcheries just like spitting them out. They're sort of like, there's lots of them in the streams of the Sierra Nevada, for example, and just, but yet they're the same fish. I don't know. I just find the whole thing really fascinating. I was just wondering if that comes into play when thinking about management via genetics. But
yeah, well, and what was the name of the person?
It's called Species Complex was the name of the article.
Okay.
It's colon something, and Peter Alagona.
Okay.
He's at UC Santa Barbara. Yeah.
Yeah, I mean, a point that I missed from your question before or hadn't thought of was, I think what you're getting at is the ways in which like the context really determines the value too of the species.
Right.
Like rainbow trout, here in Colorado are this, in some ways wanted as a game fish, but also kind of devalued because they're not native, and yet, or in inland watersheds in California, the same versus the coastal rainbow trout, which is valued.
Precisely, yeah.
So there's a great book called An Entirely Synthetic Fish, all about the history of rainbow trout. So highly recommend.
All right. Anybody has a question, because I do. I don't want. I'll give you.
I have a question.
Go ahead.
Thanks, Christine. This is really interesting. I have two questions that are a little different. One, I think one of the things that was so effective about your presentation were the graphics. And I'm curious about the drawings, paintings of the fish and where those came from. So that's the first question. And the second question, I'm interested about the reactions of the people you've interviewed, the conservation professionals, and how these questions that you're bringing up and sort of interrogations, how the responses have been.
Yeah, okay, to answer your second question first, I think the conversations thus far have been very productive and constructive, and there are these I would say there are these like, many of these concepts and these tensions are not lost on conservation professionals or scientists, but there is, in some places and in some professional contexts, a kind of concern about expressing reservations about reliance on genetic knowledge. So an example, and we have had people voice this in interviews, but another example would be in a workshop that was held in maybe, I can't remember the year, 2015 in Colorado, all about greenback cutthroat trout management. The responses to an anonymous survey of conservation researchers and professionals really illustrated some of these tensions that didn't seem to come up when names were attached, but when anonymized, there was this kind of this sense of, or these concerns about some of the potential impacts of relying solely on genetic information. Also, I think, I mean, you know, our goal in this research is not, we really don't want to come into conservation and say, you're doing it all wrong, or, you know, stop what you're doing. We know better, because, you know, we don't. We're learning as we go. And we're also working with, so from the get-go in this project, we've been working with Colorado Parks and Wildlife, which I think really helped us develop the project and helped set the stage for kind of a positive relationship between us and some of our research subjects, so that there are mutual benefits to the project. And we've also been working with the Salish and Kootenai Confederated Tribes in Montana. So through just forging those relationships really early in the process, before the project was, you know, before we had developed the project, I think helped us kind of see different perspectives on the questions we were asking. And, you know, I am hopeful that our experiences in interviews will continue to be that positive, though, you know, it may not be the case entirely. And your first question, I believe, I'll have to go back. I believe that one of the images of a trout that was an art piece was by a friend of mine. And I believe I have put the sources on the bottom of those slides, but they are not all by the same artist. And I will have to double check, because I might have just put them in my comments and not on the slide. But I can get those to you if you're interested. Anna.
Excellent. Yes. Keeping a close eye on those slides, they were really nice. I agree. So any other questions? I'll give you a chance to go ahead. I'll hold mine to the very end. If not, okay, let me go. So
actually, Rafael, can I ask a quick question real quick? Do you have Christine, and forgive me if I'm missing something totally obvious here, but do you have a public, like for people who would like to say, hey, read this, do you have a publication or something that sort of captures the essence of what you've talked about here, or is this sort of new work that you're working towards?
Well, it's work that is accepted but hasn't
Okay.
yet. So I mean, I can send you a copy. I'm happy to. It'll be in Science, Technology, and Human Values.
Oh, right, that's right. Okay, no, that's great. I can wait. I just I hope something's coming soon.
Oh, it's coming.
Awesome.
all right, perfect. Any other comments or questions? All right. Oh, Brian, go.
Oh, actually, just a comment, not really a question, but Christine, thank you so much for the presentation. Very eye-opening. And I agree with many of the comments.
You're getting caught, Brian. Maybe you want to turn off your camera.
Brought up Idrin. I don't have children of my own, but I work a lot with them. And as you're, okay.
If you can, yeah, barely. Can you turn off your camera?
Actually, I think, Raphael, it's probably coming from you, because I think he's coming through pretty clear for most of us.
Oh, really? Then it's just me. Ignore it.
Oh,
Great, absolutely.
no.
Go ahead, Brian.
Yeah, that's okay. So, Christine, I was going to say, just comment quickly that as you're describing sort of the differences between phenotype and genotype, I was just thinking to myself that it helps me appreciate how difficult it is to communicate what that is to a child. The idea that two fish can look the same. And in fact, some experts can't really tell the difference between hybrid versus native cutthroat trout. But how do I explain to a child that one fish is going to be killed because it's not the right fish, or it's not as good as the other fish? How do we as scientists, I guess the question I ask myself is, communicate the relevance and the validity of the work that we do to folks who are in situations where they see a fish and they understand what it does in that stream, but perhaps it's trying to make sense of why we're killing off a whole bunch because they're not genetically right. And it just helps me understand the challenges that we all face. Myself in particular, when I do the work that I do, I take for granted sometimes that it's understood. But it's your talk has really helped me appreciate the nuances involved. And I need to go back to my own drawing board to figure this out. So I really appreciate, that was just my comment. Thank you so much.
Thank you, Brian. I feel like I don't know about the rest of you, but I certainly learn a lot and think about things differently when I try to explain things to kids, my own included.
True. Explaining something is the best way to learn it, right? That's what I tell the students. All right, so I mean, again, it was an awesome presentation touching in so many highlights and so many important points. I think many of them, and I completely agree with the ones have been brought up. I think we're all on the same channel. It just reminds me several things that you mentioned. We have a student that just finished his honors thesis doing an analysis of the adaptive management process carried out by the Forest Service and the role of social learning and how social learning contributes to the management process, which is something that, like Gregory was mentioned, is very, very interesting, the work you're doing of how to bring the hardcore abstract science, some people would think abstract science, onto informed decision making and start changing values, beliefs, and behaviors, right? Which are core to the other comment that Lisa was talking about, ways of knowing and indigenous knowledge and traditional ecological knowledge. So many points that we're very excited about your presentation. But one thing that reminded me as you were talking was the work by Bill Janowski in the Forest Service, I believe it's Region 2. And is in your interaction with managers, what would you recommend, like for our students and ourselves, the faculty, what would you recommend will be the best ways to get across the point and start affecting the informing management activities and changing values and behaviors?
I mean, I can't answer that because I don't know. And I would say the way that we have gone about this project has really been not in trying to influence managers, but in trying to learn from and with them. So while in this presentation and there are some kind of ideas and arguments that I'm making, I would say thus far, it has been more a process of learning. I've tried to come in and learn from others that I have spoken with and worked with, and there hasn't been that sense of, we haven't really been trying to inform managers of kind of anything yet in this project. Certainly we are sharing the work that we are doing, and the work that we're doing is being shaped by managers, but I feel like I'm not the person to be able to provide a strong answer to that question, Raphael. I apologize.
This is good. It's just, again, I think it's very interesting to think of trying to make the science more relevant to how things are conducted, right? All right. Any other questions or comments? We're getting close to the time allocated, just to be respectful to everybody's time and to Christine's time. No?
I just say thank you very much. Thanks for the thoughtful questions. They have caused me to think about things I hadn't previously, and I've learned a lot from your comments and your questions. And if you have anything else, any resources that you'd recommend, or any other questions or comments, please feel free to send me an email. I always appreciate feedback. And thank you for this opportunity. Really fun, really nice distraction.
Thank you so very much, Christine. We really appreciate it.
So much.
Thank you so much. Very interesting. And I think all the students and all the faculty that could not attend today will enjoy the recording.
Thank you so much, Christine. This is a highlight.
Thank you.
Great.
Thank you.
Thanks.
Thanks.
And bye, everybody.
Bye, everybody.
All right.
Thanks again, Christine.
Bye.
Bye and thank you.
Bye.